Now, since we are discussing the various methods of antibody assays, the antibodies estimation, so in this module, I will discuss another important method which is used, which is called as Enzyme Immunosorbent Assay. That how we can make an assay of antibody by this method. Now the initial amino assay formats, they described were the methods based on the agglutination reaction. That is, what happens when antibody and antigen, they are just joined together, they are added together, there occurs the agglutination. Agglutination means that there become the precipitation, and we can assay from the degree of agglutination that how strong the antibody is present over there. Now, for example, in this case, you can see in the diagram that the antibody excess, if there is antibody excess, there is no agglutination. No agglutination occurs. If agglutination of at the point of equivalence, that is, they are the equal, then the agglutination occurs. That the precipitation, it occurs, and we can see it. And then there is, and if the antigen, on the contrary, is in excess, again, there is no agglutination. So we have to adopt a middle pathway that when they both are equal, agglutination will occur, and that will render us some method to assay them. The reaction between antibody and antigen, that can be observed when the agglutination occurs, as I have described earlier, that by agglutination is a method to determine the concentration of antibody or the concentration of antigen, vice versa. Now, the vast majority of immunoassays, they are carried out, they fall into the category of enzyme immunosorbent assays. That is, enzyme immunosorbent assays, that is a vast majority for the methods for the assaying the antibody concentration. Now, what happens? That routinely used for the diagnosis of infectious agents such as viruses and other substances in the blood, this method is used, and the antigen which is present is the substance or the agent to be measured. So during this method, for this method, we measure the concentration of antigen which is due to the infectious agents, that is the viruses or other diseases cause elements, and that is measured over there. Now, immunosorbent assay, which is actually in the brief is called as ELISA. And the most commonly used solid phase is the enzyme-linked immunosorbent assay, ELISA. ELISA means enzyme-linked immunosorbent assay, which stands for ELISA. And there are ELISA plates, that is the solid support. And immobilization on the solid support is achieved by the use of coating antibody. Antibody is coated on that plate, and that antibody which actively traps antigen to the solid plate. So that antibody is coated, and the idea is that when it is coated, the antigen will be absorbed on that. Now, there are A second antibody, which is labeled with a reporter enzyme, is allowed to bind to the immobilized antigen. So the second enzyme, which is known as the reporter enzyme, is allowed to bind with the immobilized antigen, and the enzyme substrate, what it is then added to the antigen antibody enzyme complex, which is on our solid plate, and a reaction occurs. There are many permutations of this method, but all of them rely on the antibody antigen complex being formed and the presence of it being confirmed by the reaction of the reporter enzyme. Now, reporter enzyme is an important component of this reaction. The antigen is central to the assay as it provides the bridge between solid phase and the signal, which is signal generating molecule, which is the antigen, and that is to be measured. The coating antibody also concentrates the antigen from the sample as it binds the antigen irreversibly. So you see, in this method, a solid plate is used on which the antigen reporter enzyme and the immunosorbent complex that is formed, and that is measured by the agglutination, and as soon as the antibody concentration increases or decreases, then the antigen from the sample, it binds the antigen irreversibly, and then it can be read from the ELISA machines or other component. So you see that previously we used spectrophotometry or other method to determine the concentration. But here we are using a solid phase where the reporter enzymes are connected or attached to the molecule, and the antibody and antigen that can be measured irreversibly and precisely in this method.